But speaking of continuing conversations, it's really time to move on to our colleagues in IITA. To those of us who are just joining, good morning, good afternoon, and good evening, wherever you're joining from. You're very welcome to this Marathon Spotlight event, which is taking us on a 13-hour journey around the world. Today, we're joining all of those who are celebrating the International Day of Women and Girls in Science. And we in CGI are taking the opportunity to celebrate our own fantastic community of women scientists who, as we've heard throughout the day, are really leading the way in a wide range of research and scientific innovation, very clearly breaking barriers and finding new ways to advance the transformation of food, land, and water systems in climate crisis. For those of you I haven't had the pleasure of meeting before, my name is Fiona Farrell, and I lead CGIR's function that works to advance gender equity, diversity, and inclusion in our global workplaces, or GDI for short. We in the GDI function, we just celebrated our first birthday. We were launched in January 2020, and we have a very clear mission, which is to work with leadership and management and staff across CGIR to make sure that our workplaces are truly enabling and inclusive for our over 10,000 people. Our goal is to ensure that diversity in all its dimensions is embraced, and that everybody in our workforce is really supported to reach their full potential. For those of us who are working in this area, we know that it's not only the right thing to do, it's the smart thing to do, because we know that diversity can bring the different perspectives and the innovation we need to deliver on our critical mandate. The work of the GDI function, many of you will be aware, is guided by our GDI framework and action plan. If you'd like to learn more about these important documents, what we do and how we do it, access any of our knowledge hub, our training, our guides, you can learn more by going to the GDI webpage, and you can see the details up there on the screen now. Among the many initiatives that we've had the honor of supporting in 2020 was the launch of a new employee-led resource group called WIRES, Women in Research and Science. And WIRES brings together all of us in the global community who seek to support our incredible women scientists, providing them with a supportive, safe space to meet and to learn and to grow. And today's event follows on closely from the official launch of WIRES on the 21st of January. Today, we recognize the significant contribution of women scientists by showcasing a sample of their exciting and innovative work. And as we continue on with IITA on our 13-hour-long journey, we remind you of a few housekeeping rules, as you can see on the screen right now. We invite you to submit your questions and your comments via the Q&A box, and to reach out to Thomas if you have any technical difficulties. We also invite you to join WIRES using the contact details on the right-hand side of this slide. And it's on this note that I say congratulations to our Alliance colleagues, and it's with my great pleasure that we welcome the speakers for our 9th Power Hour today, which is being hosted by IITA. Handing over to you now. Thank you.
Thank you, Fiona, and good morning, good afternoon, and good evening to everyone. My name is Sylvia Inola, and I'm speaking to you from IITA Ibadan, which is our headquarters. We're going to start our presentation with a welcome address by our Acting Director General for Research and Development, Dr. Bernard Van Lau. Over to you, Bernard.
Yeah, thanks, Sylvia. Thanks a lot, and really many congratulations to the organizers of this event. I'm seeing currently we still have over 170 participants online, which is really, really encouraging. So well done, and looking forward to see results of this discussion. Now, I wanted to make 3 points. I was only given 5 minutes, so I need to focus on probably 3 points. And I'm pretty sure some of those points have been raised already today, but often people say it doesn't hurt when you repeat a good message twice or 3 times, I guess. So the first point, and I just heard allusions to this already, diversity in research leads to diversity in science solutions. I think there's enough evidence of that, more and more evidence created, and there's a number of ways in which that can happen. Of course, it's about having access to the best talent to accomplish your research, We also know that diverse organizations and teams, they usually use a greater variety of perspectives. The sort of solution spaces for diverse teams is usually more complex and more addressing complex problems that we are all facing on, of course. And thirdly, more diversity leads to a better understanding of the context and the environment for which we are working and for which we are trying to find solutions. So we need women in science, we need men in science, we need older scientists, maybe somebody like the one who's currently talking to you. We need younger people. We need people from different backgrounds. We need people from different upbringings, and so on and so on. The second point I wanted to make is it's good to talk about the need for gender in R&D, but we need to start at home. It's good to talk about it, but we need to start at home, within our own organization. As I was introduced, I'm working for IITA. We currently have 23% of our scientists that are women. Is that a lot? I don't know. It's 23 is better than less than that figure, but it's not 50. So I think we can do better. And I think today is a day where we can actually put this in perspective, work with the GDI initiative to see what we can do as an organization to do better ourselves. We have to. There's of course reasons why this number is 23 and not 45. I think we know a lot of those reasons, and I think via the GDI initiative we can actually address some of them and get at the figure we should get at. And the third point I wanted to make is that a science career can be a very rewarding career for women. I think we'll show clear examples of that. I also just understood that having or raising children is not a bottleneck. Let's keep that in mind when we talk about women and gender in science. You will see a lot of examples of that from colleagues, women scientists, from a wide range of disciplines. I think we'll have people from the breeding area, seed specialists, pathologists, and so on, tissue culture specialists, food and nutrition management, and so on. So we'll provide evidence of that. You'll see that those women also come from very different backgrounds and have worked in different countries. Some of them have been for a longer period at IIT with leadership positions somehow. Others have been or younger and have just joined. And I think you'll all see examples of that. But more importantly, we are not only trying to work with women scientists that have already a degree, that have been trained, but we are also trying to invest in attracting younger people, students, into science careers. In IIT, we do have our STEP program. STEP means start them early, where we try to work with secondary schools and let young people be interested and incited by an agricultural career. We have our youth program, our Enable Youth program, which is also trying to interest women and girls into following a science career in agriculture. And of course, IIT's women's group is working closely with the RAGA program, Raising Girls' Ambitions in Agriculture. So these are the points I wanted to make. I think my 5 minutes are finished. So looking forward to this discussion, and back to you, Sylvia, for introducing the colleagues that we are very proud of today. Thank you.
Thank you, Dr. Bernard, for a very interesting introduction and for the support. Our next presenter is Lina Tripati, who holds a leadership position as the director for R4D in Eastern Africa. She's based in Dar es Salaam, and her presentation is going to be on genome editing for crop improvement. Over to you, Lina.
Thank you very much. Hello, everyone. So I will briefly talk about what genome editing can do for crop improvement. Next slide, please. So biotechnology has a lot of potential to transform agriculture, particularly when the population is increasing and which demand for more food with the limited resources. And also, the climate change is actually adding a challenge to that. So that means, like, we have to look for the sustainable intensification in order to double the crop production. And when we talk about sustainability, it cannot come only with the conventional technology or only with the biotechnological application. So what I'm saying is we have to work together. We have to use all the tools which are available in the toolbox, whether they are conventional technologies or biotechnology, and apply all of them in order to increase and have the better nutritious crops. Next slide, please. So genome editing has a lot of potential for crop improvement, and because it is a very precise tool which can efficiently make specific changes in an organism's genome. But genome editing is not new. Nature has been doing editing in genome for a very, very long time. When farmers started using agriculture, those times the nature was editing and then farmer was selecting. That's how they were getting the improved varieties. But in 20th century, the mutations were accelerated using chemicals and radiations. And later on, the technologies were more precise technologies were developed using gene targeted mutations like zinc finger nucleases, talents. And recently, CRISPR has become the most popular genome editing approach. This technology was first reported in 2013. And I'm very happy to say that, in 2020, two women scientists, Jennifer Dauda from UC Berkeley, and then Emmanuel Carpentier from France actually got the Nobel Prize in chemistry for this technology, CRISPR technology. So at IITA, we are applying genome editing for several traits. We are using it for functional genomics, for the gene discovery, for the nutrition enhancement, for the yield improvement, but most effort are on the biotic stress for developing disease resistance for bacteria, viruses, and fungi. And we are also planning to apply for abiotic stress like drought, and then herbicide tolerance like the weed control. Next slide, please. So what we are doing in my lab is we are transferring the knowledge, what we have generated from the wild type of banana species into the cultivated varieties. So wild type of a species, that those bananas are seeded bananas, so they are not edible and farmers doesn't grow them. But then the most of the cultivated varieties in bananas has a lot of issues with the diseases. And it's not only one disease, there are several diseases which are present actually in the same field at the same time. So, we are targeting, even generating the varieties which have resistance to multiple disease. So we are taking this information which we have generated using different molecular tools from the wild type varieties and transferring them to the susceptible variety. So we are not bringing anything outside of the banana. It's just the bananas that, we are improving the bananas based on the information. So we are either overexpressing the genes, defense gene for developing the resistance, or we are actually knocking down the genes which are responsible for the susceptibility of those genes for the diseases. Next slide, please. So some of the key take-home messages are that application of biotechnological tools has potential to transform agriculture. When I said biotechnological tools, that's not only genome editing or transgenic, that includes molecular markers, tissue culture, even molecular diagnostics. Those tools has a lot of potential. Genome editing tools are becoming popular molecular tools of choice for crop improvement as well as the functional genomics. CRISPR has rapidly become the most popular genome editing approach due to its simplicity, efficacy, specificity, and also, as I said, it can be multiplex. So, you can develop resistance to multiple diseases, and it's quite easy to adapt. So if you have a tissue culture transformation lab, you can easily adapt this technology into your lab. Now, another advantage of this technology is that the genome edited crops with no foreign gene integration, that means there is no foreign DNA from anywhere outside of that plant species, are not regulated in several countries, and that's what we are hoping in some of the African countries will adopt too. The application of CRISPR-Cas-based genome editing has been demonstrated at IITA for disease-resistant bananas, but we are also using it in cassava. And we are building a capacity of young African researchers. In my lab, so far, we have trained almost 30 master and PhD students, and actually about half of them are female scientists, the young researchers. And we also provide a lot of short-term training to the African researchers and then help them, setting up their lab for doing this type of technology. Thank you very much.
Thank you very much, Lina, for breaking down your work in 3 short slides. I know it's very complex, but thank you so much. Our next presenter is Busi Madzia Dixon, and she heads the Food and Nutrition Science Unit here in IITA. She's based in Ibadan, and her focus of recent has been nutritional assessment, which promotes agricultural nutrition and health linkages. Her presentation is titled, "Why is it important to know the type and amounts of food consumed by different groups in a population?" Over to you, Busi.
Thank you, Sylvia. Good afternoon, colleagues. Good afternoon, good morning, wherever you are in the world. As Sylvia says, I'm a CGIA Senior Food and Nutrition Scientist working with the International Institute of Agriculture, and I'm based in Ibadan, Nigeria. And I think I may be the only female scientist who have been in the CGIAR for over 20 years now. So I'm very proud of that. Don't ask me how I got where I got and how. Now I'm going to just talk about how it is important that we know what people are eating, the types of foods that they are eating, and the amounts of food that they consume. Now, we all know that in the world, we are faced with 500 million of people who are obese. We have a growing population of 7 billion people. And then we have 800 million people who are chronically suffering from hunger, and 2 billion are malnourished. And so then globally, we are faced with a triple burden of malnutrition, whereby we have undernutrition, we have overnutrition, and we have micronutrient malnutrition. I'm just going to focus the presentation on an emerging problem, which in developing countries, we still look at it that it is for developed countries. And that is the problem of obesity and underweight. Can you please go back to the slide? I will let you know when. And this problem of obesity can lead to cardiovascular diseases, diabetes, cancer, and respiratory disease. These diseases are grouped, these conditions are grouped together as non-communicable diseases. Now, the global panel on a disease study indicated that most of the diseases are actually related to diet. As you can see in the bar chart I have here, all the green bars, those are diseases which are related to dietary risk. And so then because we are what we eat, it becomes very important that we know what people are eating, how much of it they are eating, and then we also need to know, collect indigenous knowledge on processing and utilization of the different food crops which are available within the communities. We also then have to look at the nutritional quality of the raw foods and also those processed foods, because this is the information we need to actually go ahead and have targeted interventions. Next slide, please. Now, how do we use this information, or how is this information used? You know that once we know what people are eating and how much of the foods they are eating, then we can use that information to assess the adequacy of the nutrient intakes which are supplied by the foods they consume every day. We can also use that information to assess how they adhere to guidance. We all know that we have the dietary guidelines where we tell people that eat so much of this per day, eat so much of this per day. So such information will allow us assess adherence to those guidelines. It will also allow us to develop consumer guidance, teach consumers which foods are good for their health. And then it will allow us to know what actually people are eating and how much of it are eating. And this will feed back into agricultural policies and programs, food and nutrition policies and programs. And then it will also help scientists to target and governments to target their programming interventions. And so then what does it take to be a female scientist in the CGIAR? First of all, you need to know your stuff. That means you need to know your work. You need to be good at your work. And then this, you bring in on top hard work, you bring in assertiveness, you bring in perseverance, you bring in self-confidence. You have to be competitive. You cannot survive in the system if you are not competitive. And of course, you have to be articulate. Thank you very much.
Thank you, Busi, for a very interesting presentation and calling attention to something that a lot of African countries do need to. Our next presenter, we're moving on to Uganda now, is Valentine Nakato. She's a plant pathologist with IITA Kampala, where she leads and organizes and plans banana pathology activities in support of the banana breeding program. Her presentation is on fitting the puzzle pieces within the wider world of plant science. Please, can we go back to the first slide?
Thank you very much, Tunde. I'm Sylvia for the introduction, and greetings to everybody from Uganda. How do we fit pieces of the puzzle? Everybody knows that science is like a jigsaw puzzle. Most of the times you have some of the puzzle pieces with you, and other times the missing pieces of the puzzle are with someone else. So you are either a mentor or a mentee, you are writing a proposal, or you're thinking about experimental designs or setting up experiments. And all these pieces of the puzzle lead to an accomplishment. To me, my accomplishment was that for the last over 10 years that I have been with IITA, my puzzle pieces came together through the attainment of my master's and PhD degrees, and ultimately the appointment as a postdoctoral fellow with the same institution in Uganda. So for my presentation, I would like to highlight two key achievements of my academic journey. Next slide, please. So during my PhD study, we developed a multi-locus variable number of tandem repeat analysis, which we called the MLVN19 scheme. And this is a PCR-based genetic typing method to explore genetic diversity of Xanthomonas versicolor musacearum populations, the pathogen that causes banana Xanthomonas wilt in Eastern Central Africa. Now, this study determined that this scheme is sufficient to explore genetic diversity of XVM, and these findings are helpful in addressing evolutionary and ecological questions that have not been fully addressed previously. These research findings were presented during the Research for Development Week at IITA in 2019. And I am happy to say that our poster took first prize in the best poster presentation competition. And for this work, I had mentors from IITA, Bioversity, CIRAD in France, University of Pretoria in South Africa, and University of Exeter in England. And I was simply the mentee with a topic who carried out the associated experimental work with their guidance. Next slide, please. The second highlight was the identification of the existence of resistance to banana Antomonas wilt within Musa Accuminata. Now, these findings revealed potential to develop banana and plantain varieties with tolerance to Xanthomonas fascicola musasiarum, and also led to doubling of efforts in developing of banana varieties that are resistant to banana Xanthomonas wilt. In fact, this publication was among the top 10% most downloaded papers published between January 2018 and December 2019. Next slide, please. So for my postdoctoral research, I'm engaged with two projects, the accelerated breeding of beta bananas and the AFLASAFE projects. And for these projects, I'm both a mentor and a mentee in the sense that I am mentoring young scientists within my team as I continue to be mentored by other senior scientists. With the accelerated breeding of beta banana project, our contribution as a team is towards enhanced host plant resistance to major pest and disease constraints through improved pathogen identification and accelerated early stage screening of resistance. And this will facilitate early decision making by the breeders, but also improve the efficiency of the breeding pipeline. For the AFLASAFE project that is yet to start, our aim is to provide safer food and ultimately more profits to the farming households through development and deployment of aflatoxin biological control products, in addition to the integrated aflatoxin management, and we make these accessible through private sector engagement. Now, some of the challenges that are faced as a female scientist is that my ambitious nature sometimes comes off as being bossy or too assertive, and most people think that this is quite unfeminine. And I aspire to become a well-rounded contributor to the global agriculture research community that is leading to poverty reduction in the world. Thank you very much.
Thank you very much, Valentine. This was a great presentation, and thanks for continuing being assertive. Our next presenter is presenting from Lusaka, Zambia, and she is Julieta Kello, and she's a plant pathologist who conducts surveys to determine the prevalence and severity of diseases and mycotoxins in Southern Africa. She will be telling her science career story in The Rise from a Research Technician to a Highly Competitive CGIAR Scientist, What a Journey. Welcome, Juliet.
Thank you, Sylvia, for that lovely introduction. Happy celebrations to everyone out there. The issue of food insecurity and malnutrition has actually been with us for a while. And for me, my thirst to find solutions to end hunger and malnutrition, especially in Africa, started way back in the 90s. During that time, I was just a young girl in northern part of Uganda. Unfortunately, we had like rebel activities within that region. And when I was abducted, that is when I actually realized or became aware of what it means to go hungry or on an empty tummy. When I got out of that ordeal and returned back to school, I learned that actually, like my colleague, Dr. Busi said, millions actually go hungry or become malnourished, primarily sometimes because they have limited access to sufficient food or unsafe, or they're eating unsafe foods. Unlike me, that was forced by the rebels to actually go on an empty tummy. Most of our farming communities actually face food insecurity, primarily because they are fighting very small microbes or insect pests and diseases that not only reduce their crop yields, but also reduce the quality. And ultimately, somebody will have less available food for consumption or for even sales. Since then, I actually channeled all my energies towards fighting pests and diseases using biocontrol approaches that farmers can actually have better yields and safer foods. Maybe to divert a bit, initially I actually studied a course that I was supposed to be a very good technician in medical laboratories or Bureau of Standards, but I knew this would not actually help me to realize my goals of contributing towards hunger reduction. So I switched to agriculture, climbed through the ranks and became and did my PhD, and that is what led me to where I am. So in 2013, I joined IITA, and one of my tasks was actually to address the issue of ear rot diseases. So ear rot diseases are mainly caused by fungi or mold that infect the crops when they are still in the field and sometimes continue right into storage. Apart from reducing yield by almost 50%, they also produce toxic secondary metabolites in infected grains. And some of these actually reduce the safety or the quality of the grains. When consumed, especially some of the mycotoxins or these secondary metabolites, especially aflatoxins and fumonisins, they are actually harmful to us humans as well as to our livestock. Unfortunately, the exposure to these mycotoxins are really, really high across Africa, and exposure has been linked with the poor child growth, weakened immunity, liver cancers, and even consuming very high levels can be lethal or causes death. Over the past decades, IITA, in collaboration with the national and several international research centers, actually discovered that there is a safe way to control one of the mycotoxin problem in the maize and granite value chains, and that is aflatoxin. And the product that I'm a proud member that I contributed to developing is called Aflasafe, a biopesticide that is capable of actually reducing aflatoxin contamination in maize and granite value chains by over 90%. I will not delve into the details of how we have produced this product, but for those who want more details, I've provided actually the website on the screen. The main challenge of some of the research that we do is that sometimes we deal with invisible problems. Mycotoxin is one of the invisible problems. You cannot see a mycotoxin with a naked eye. And as such, actually, most people don't take you seriously when you're talking about that we have this huge food, unsafe foods in our communities. People don't take you seriously. And because of that, the registration process for these biopesticides within the region was really slow. Primarily, some of the nations or some of the countries we operate in lacked the necessary legal framework for registering biopesticides. That said, I'm proud that with the fellows have registered 3 Aflasafe products within the Southern African region, that is in Zambia, Malawi, and Mozambique. And with such a product on the market, we can actually be proud and say that the health and livelihoods of millions of farmers actually will be safeguarded. Next slide, please. Next slide. Next slide. Thank you. So the other challenge that we are facing within the Sub-Saharan Africa is actually the issue of striga weeds that can reduce yield by even 100%. Most of our farmers, if you interact with them, those facing such a challenge, will tell you that no matter what they try to do, they are actually not finding a solution to get rid of a strider, primarily because we are having poor soils. Even the issue of climate change is coming in, and that is why we are having heightened strider weed infestation. Some farmers actually end up abandoning their farms because of this. And together with a group of researchers in the US, in Europe, and even here, African scientists, we have teamed up to find a biocontrol approach that will be environmentally safe for our farmers to use in mitigating the aflatoxin, the strider weed infestation, so that they can get better yields. Next slide. So in my journey, that has brought me this far. I had really the help, or I passed through the hands of very many mentors and great supervisors from IITA, from Germany, you name it. And most recently, I passed through the hands of a word that empowered me. And under this umbrella, we have formed the Zawad cohort here in Zambia. And through this, I really hope that I will inspire as many or thousands of young girls to actually embrace science and technology in their career paths. What we have realized is that these young girls, they think that science is too difficult for them, or agriculture or farming is for the poor. So I really encourage each and every one of us scientists to encourage these youngsters so that they don't really lose their dreams of becoming great scientists. Let's encourage them, let's nurture them. With that, I want to say thank you for your attention.
Thank you so much, Juliet, for such an inspiring story and for inspiring most young girls that are out there. Our next presenter is Sika Begbelegbe. She's an agricultural economist based in IITA Lilongwe, Malawi, and she focuses on using bioeconomic modeling tools for foresight analysis. Her presentation is entitled, "What Does the Future Hold? Science Can Help." Over to you, Sika.
Thank you, Sylvia, for the introduction. Indeed, my name is Sika Begbelegbe, and I am a CGIAR scientist working with IITA and based in Malawi in Africa. The title of my presentation is "What Does the Future Hold? Science Can Help." Next, yes. Some of the work that I do in IITA is related to making projections on the future of food security, nutrition security, and poverty in Africa and also across other regions in the developing world. Here I'm presenting some of the work that I've been doing recently with my colleagues. We all know that Southern Africa was cited among the climate change hotspot by the IPCC. And also future projections are implying that the region is going to experience drought in the future that will be lasting longer and also that will become more intense. So my team wanted to be able to quantify the impact of these future droughts on food security in Southern Africa. And basically what we found is that with the future droughts that could occur in the future, maize production will be more affected negatively compared to other crops that are grown in the region. And that would translate into severe consequences on food security in the region, despite having an improvement in incomes, despite having economic growth up to 2035. The key reason for such a result is that Southern Africa is heavily dependent on maize as a food crop. So what we did with my team is by using the results from this work, we were able to come up with some policy recommendations for policymakers in Southern Africa. Next slide, please. So you will see some of the work that I've done has been published in very good scientific journals, as shown here. And one thing I want to highlight is that last year I had the opportunity to be invited to a webinar that was organized by Egerton University in Kenya. The webinar was on the theme of demazing Kenya. So basically finding ways of reducing the dependence of Kenya on maize as a food. So that platform brought together scientists and policymakers, and we had to share our insight, our ideas with one another. So this platform gave me a great opportunity to be able to share the unbiased findings from my research with policymakers. Now I'll talk about my aspirations. I have two of them. The first one as a scientist is that we should have better relationships between climate change scientists and policymakers in Africa. Everybody remembers, I think, what happens in 2018 in Cape Town, a city that was running out of drinking water because of 3 previous years of consecutive drought. If indeed in that city there were better relationships, better connections between the scientists and the policymakers, the city would have been able to better manage the situation they faced in 2018. My second aspiration as a woman scientist is that indeed we have more women scientists working in science in Africa and also in other parts of the developing world. And to do that, what we need is to be able to mentor our young girls in primary school, high school, university, and help them to become the scientists that God wants them to be. Thank you.
Thank you so much, Sika, for giving us an insight into the future through your work. Our next presenter is Delphine Ama. She's a plantain breeder based here in Ibadan. And she leads the plantain improvement initiatives in Western Central Africa, and she builds capacity for plant propagation, micropropagation, and macro propagation. Her presentation is entitled Plantain Breeding in IITA, Turning Challenges into Opportunities. Over to you, Delphine.
Thank you, Sylvia. Greetings, everyone. I'm delighted to be here to talk about my work. My inclination towards science began in my early school days, where I was passionate about forms of life, how they coexist, and how this knowledge comes into play to improve the quality of life. I nursed this passion throughout my academic career into my professional career, where I now help in creating and transforming scientific innovations that address food security issues for Africa, particularly for plantain, an important crop for Africa. When I say plantain, what comes to mind? Plantains are starchy types of bananas that are mainly consumed when boiled, fried, roasted, or transformed into flour for making other culinary products at different ripening stages. Although the crop is consumed worldwide, it is mainly produced in tropical and subtropical regions of the world. In Africa, where over 50% of the plantains in the world are produced, these are grown in the humid tropics, in the humid regions of West and Central Africa. It is estimated that 3.2 million households depend on this crop, as it is mainly grown by smallholders. These growers face several production challenges, the most prominent, which are pests and diseases. Diseases like black sigatoka, a fungal leaf spot disease, pests like nematodes that affects the culm and weakens the stem and cause the plant to topple, leading to sometimes up to 100% loss of yield. Another major challenge is the lack of seed, because the plants are propagated through suckers obtained from the mother plants, which are side shoots. Now, these challenges present themselves as opportunities for improvement. Hence, we develop solutions to address these challenges to create better opportunities for the growers who grow this crop. Next slide, please. We do breeding to generate improved varieties that combine resistance to these diseases and pests, but also include other desirable characteristics that are preferred by growers and other end users of these crops. And also we focus on some characteristics that add value to the crop in terms of nutrition, such as provitamin A, which is significantly high in some banana types. To do this, we explore the banana diversity that is available worldwide and use this diversity to make crosses, to combine and recombine all these interesting characteristics so that we can have good hybrids for farmers. Plantains are normally seedless, just like other bananas. But when we make these crosses, we do get seed. And when we are fortunate to get seed, we extract the embryos and nurture them in vitro in tissue culture, and then let them grow into seedlings, and we nurse them into plants. Then we evaluate these plants in the field over several cycles to select the best hybrids that combines these desirable characteristics so that we can deliver them to farmers. Next slide, please. I also work on ways to propagate. Next slide, please. I also work on ways to rapidly propagate planting material to make available good quality seed to farmers.
Go back, please.
I'm very passionate. Previous slide, please. I am very passionate about plant tissue culture, and I bring on my technical expertise on this area to build capacities in Africa, particularly for labs who use these techniques in micropropagation. I also train people on macro propagation, which is a low-cost technique, that is more practical for growers and entrepreneurs in community settings. I am quite intentional in collaborating with other stakeholders to improve the efficiency of all these techniques. Next slide, please. I must say that as a woman in science, I am very fortunate to be surrounded by fantastic women and men in science who have been incredible sources of support. I aspire to continue on this journey, transforming challenges into opportunities, particularly for smallholders, and to keep climbing up the ladder. And as I do this, I see myself not just as a woman in science, but as a person in science who has what it takes to do a job that she has committed to do. I will end here with a word of encouragement to other women in science by saying, know yourself, believe in your own unique values, and keep empowering each other so that you can be an inspiration to the next generation. The world definitely needs your talent. Thank you very much.
Thank you so much, Delphine, and thank you for showcasing your work, and good job with the plantains. My next presenter, which will be our last presenter for this session, is Mercy Lehodieberujo. She is a seed specialist focusing on clonal propagated crops, and she will be speaking on technologies for rapid multiplication of clonal crops. Over to you, Mercy.
Thank you very much, Sylvia, for the introduction. And I am super excited to talk about the work we do here at IITA and how it has made so much impact in the lives of farmers. Growing up, seeing farmers working so hard and yet and so little has always been a thing of concern for me. And as I was growing, I was like, how can we make this possible to ensure the farmers actually get the worth of all they're putting into what they do? A huge aspect of the food security actually depends on what we do in IITA, which is what seed production, seed supply, and modeling seed systems. A modeling seed system for Nigeria and so many other African countries. And talking about Nigeria, we have most of our staple foods are clonal crops, which are cassava and yam. And so far, they had so much challenge in terms of production, which is the low multiplication rate of these crops. For cassava and yam is a very tough one. And over several years, this low multiplication rate has been what has been used to produce the planting materials for these crops. However, we needed to look through, we needed to explore, because we have varieties that are bred every year, every time, year in, year out. However, these varieties are not adopted, and it is not because the farmers do not like them. is just because the materials, the planting materials are not accessible. They are not available. And this is because of this problem of low multiplication rate. So we had to explore technologies to ensure that these materials these good varieties are rapidly multiplied to ensure that farmers get them in good time. And one of which is the semi-autotrophic hydroponics, which we use for cassava stem propagation. So far, it's been very successful in our system here. We have adopted here at IITA since 2016, and it has helped in the rapid propagation of in vitro plantlets under controlled environmental conditions. However, it's not as complicated as the in vitro, the tissue culture one, where we need aseptic condition, absolute aseptic condition, and some capital-intensive equipment. Is a low-cost technology that we're using to multiply cassava planting materials, and these materials are deployed to the field after multiplying in the greenhouse. So in a short while, in a small lab or greenhouse, as the case may be, you can multiply as much thousands of planting materials that you need. And while we are multiplying these materials, they also have the genetic purity in place. They have their disease status very clean, and we are able to produce clean material and deploy them to the field. Next slide, please. So while we have for cassava SAH, we also have for yam propagation, the aeroponic system. The aeroponic system, like we say also for yam, the traditional method before now is the use of the yam tube by itself, where you mini-step them. And this is like making what you're supposed to eat, turning them into planting material, which also eats into what is supposed to be produced. But with the aeroponic system, you can use different parts of the yam plants, such as the tissue culture. We use the vine seedlings and all that through a technology. And these are highly rapid multiplication technologies where you can get millions of seed, and you can get different types of seed. Like for the aeroponics, you can use the vine seedling as a seed material. You can use mini tubers that also produce through the technology, also as seed material. And in a short while, you get very healthy planting materials, and these planting materials are sent into the field at the time when needed. In the face of the COVID last year, these technologies really helped us here at IITA because we didn't need to go to the field to multiply our planting materials. And this is while the COVID was on, where we had restriction in movements and all that, we went on with our work in these confined spaces, in the greenhouse, in the lab, of course, adhering to the COVID restrictions and guidelines, but we're able to produce as much as we can. And these are the materials that we are deploying and making available to farmers this year. In fact, it was a blessing for us, and we are happy that we're able to impact food security, the looming disaster that would have come post-COVID. So we have the system in place to assure that no matter what the situation may be, we'll have a way to produce planting materials for farmers when everything becomes normal. Next slide, please. So I won't talk much about the plantain because my colleague has spoken a lot about that on plantain, the micropropagation for plantain. This is also very important. And these are the things we do. And we're very glad that we have been able to provide farmers with healthy planting materials. We have also helped in restructuring the seed systems of cloner crops in Nigeria. They were not evident before, but with these technologies in place, it has served as a good model for these seed systems to take off. And we've also helped farmers increase their income through seed production and commercialization. And my message to girls out there is that is the key word, everyone has been saying it, is I think is assertiveness. When you're assertive and you know yourself, I think the sky is going to be your limit. Thank you for listening.
Thank you, Mercy, for sharing your work with us and encouraging the young girls. We have very few minutes left, so I will move on to your question and answer. We'll take just one question and then we'll move on to our closing remarks. Elizabeth, can you take a question, please?
Yeah, thank you so much. Exciting presentation for the night power hour. Indeed, these women have been fantastic. I know it's going on in somebody's mind. How can I be helped if I want to become like what they are talking about? I want just one of you to tell me, how can these young ladies be helped out there listening to us? Any of you can take it up. I want a young lady to tell her, Madam Busi gave us about 7 points that you should know your staff, you should persevere, you should be self-confident, you should be competitive, and you should articulate your facts very well. She did say those ones. But then if somebody is asking, I think I will ask our director, Lina, if somebody wants to know how we can help, what will you tell that person? Thank you.
So I will tell that person that, first they need to be determined, focused, and get a mentor as early as possible. Because, without mentorship, you cannot reach the level you want to. Because at every level, starting from the childhood up to the stage I am right now, we need mentor at every stage. So, I think the mentorship is very important. That's what one word I will say, get mentor as soon as possible.
Yeah, I think that is excellent. One person asked a question and it has to do with these young girls. So I have already asked that question for that person. Now, the last question I will ask is, what is one thing that has kept you going? I mean, for Delphine, Mercy, Shika, and Juliet, just give us just one thing. It could be a statement that has guided you throughout. Madam gave us 7. It shouldn't be part of what Madam gave. Just give me one of yours. And that ends the session on questions and answers. Thank you. Sylvia, you can take it over from there.
For me, yes, for me, Elizabeth, it's passion.
Okay, thank you so much, Sika, for giving. Our last presenter is also a co-founder of the WERF, and that's Gundula, who will be closing up this session for IITA. Gundula?
Yeah, good morning. Good morning and good afternoon. My name is Gundula Fischer. It's an honor for me to close the IITA session. The 11th of February reminds us we have come a long way with so many women at this marathon, and we need their contributions for several reasons in our research organizations. We need them because it's a basic human right. We need to provide equal equal employment opportunities, but we also need them in order to find the best solutions, using the best talent, understanding the demands of the communities we serve, and developing meaningful and sustainable solutions. So the 11th of February reminds us that we've come a long way, but also we still have to go a long way to include more women and girls, and we need to engage with girls much earlier and much more. So I would like to thank all of those who have contributed towards the organization and the content of this amazing marathon. And we are looking forward to the next session, which will be hosted by the International Potato Center, SIP. Sylvia, can we hand over to our colleagues from SIP?
I can take it from here, Gundula. Thank you so much. Thank you to, and I have to make sure I get as many names in as I can. Lina, Valentine, Juliet, Sika, Delphine, Mercy, Busi, Catherine, Gundula, Elizabeth, and Sylvia, as well as Bernard, for a really enjoyable and enlightening session. It was really humbling and exciting for us to hear just a little bit of everybody's work, and the personal journeys as well were really inspiring. It's been great to hear what has motivated many of our colleagues to take up a career in science, what sustains them, the work that's going underway, and the amazing results and successes, as well as the passion. Thank you to Bernard for his opening remarks. I agree with him. It's never a bad thing to repeat the important point that diversity makes a difference and that it adds value. And just before I finish up, I agree with Delphine when she says the world definitely needs your talent. As we get to the end of the hour, the top of the hour, I only wish we had more time with you. We truly appreciate the commitment and the time that you've taken to be with us today. I know that many other colleagues are with me when we say that we have many questions we couldn't fit in. Don't worry. This is today's session is only the start of the ongoing dialogue, and please do feel free if you're online to reach out to our IITA colleagues to continue the conversations after today's event.